Delighted to welcome you to our fifth webinar of the year. This one's got quite a buzz about it because it is the day after World Bee Day, and it's all on pollinators that live in soil. So before I welcome our presenters, I want to introduce the hosts of today, which is the British Society of Soil Science. So we're an established international membership organization and charity committed to the study of soil in its widest aspects. We bring together those working within academia, have a growing membership among practitioners implementing soil science in industry, and those with a keen interest in soils as well. So we plan to host 10 webinars during 2026. So please keep an eye out on our website, and that is where you'll find some further details. So just before we begin, some basic housekeeping. Everyone's microphones have been muted. We're using, we'll be answering questions at the end. So what we'll do is monitor the Q&A box throughout the webinar, and there may be some answered during it by text as well. So please submit any questions to the question box by 12:50. This will allow us to get through as many as we can. Also, today's presentation is being awarded BASES and NRO SO CPD points. If you're registered for either body, please contact us directly after the event. And finally, please be aware that we're recording the presentations today as well. So I want to introduce our three presenters. They are Professor Jeff Ollerton, Professor Sara Leonhardt, and Dr. Siul Ruiz. So Jeff Ollerton's an ecologist. He's also an author and consultant with more than 35 years experience studying the natural world, particularly on plant-pollinator interactions and their importance for biodiversity, food production, and society. He's published more than 90 scientific articles and book chapters, and his research has informed national and international initiatives to conserve pollinators and pollination services. Jeff advises governments, local authorities, conservation organizations, funding bodies, and the media on pollinators, biodiversity, and environmental change. Our second speaker will be Professor Sara Leonhardt. So she is Professor of Plant-Insect Interactions at the Technical University of Munich, or TUM. Her research focuses on the chemical and ecological mechanisms that shape interactions between plants and insects, with a particular emphasis on bee health and biodiversity. She has held the Professorship for Plant-Insect Interactions at TUM since 2019 and was appointed Vice Dean of Research in the School of Life Sciences in 2025. In her scientific work, Sara investigates how insects, particularly bees, exploit resources and how these interactions influence their fitness and diversity. She's also the PI of a new EU project called Prof Soil, and she'll talk about that today. Finally, our third speaker is Dr. Siul Ruiz. He is a lecturer in bioengineering science at the University of Southampton, where his research focuses on the physical and mechanical processes that govern biological activity in soils and plant soil systems. His work lies at the interface of soil biophysics, biomechanics, applied mathematics, and environmental engineering, with particular emphasis on soil bioturbation. If you don't know what that means, he'll tell you all about it. And this is the ways in which living organisms modify and restructure soils through movement, growth, and excavation. So Siul's currently affiliated with the Bioengineering Science Research Group at Southampton and holds a Royal Society University Research Fellowship. So I'm going to hand over to Jeff now to kick off the talks. Jeff, you've got 15 minutes, Sara 10 minutes, and Siul 15 minutes as well. Okay, over to Jeff.
Thank you very much, Paul, and thank you to the Society for the invitation for us to come and talk to you today. So let me just share my screen. There we go. So yeah, so I want to kick off this this session on soils and pollinators and what I describe as being a complex dependency. Yeah, by thinking about some of the statistics around the importance of pollinators. So it's estimated that something between 80 and 90% of flowering plants are animal pollinated, mainly by insects, also by vertebrates such as birds, bats, some non-flying mammals and reptiles as well. So it's probably around 308,000 species. And work published back in the early 2020s by James Roger and colleagues suggested that without pollinators, about a third of those plants would produce 0 seeds, and half of them would have an 80% or more reduction in seed production. So with those two statistics alone, we can see that plant-pollinator interactions support most terrestrial ecosystems, because most terrestrial ecosystems are dominated by flowering plants, but they're also supporting the animal populations there. The animal populations that feed directly on the plants or on their seeds, for example, or which use plants in other ways within the ecosystems. And from a calculation I made back in 2016 in a paper in Annual Review of Ecology, Evolution and Systematics, I calculated that perhaps as many as 1 in 10 described terrestrial animals might actually function as pollinators. So plant-pollinator interactions are ecologically important. They're also evolutionarily important as well, of course. They drive a lot of evolutionary diversity. But they're also important economically and nutritionally. So the high-level figure there, which is often cited, is that about 3/4 of the major crops globally, the most productive crops, are animal pollinated to some degree. And that accounts for about 35 to 40% of worldwide yield. Those agricultural pollination services were valued by a recent IPBES report at between $235 and $577 billion per year. So massive value to global agricultural pollination. Not only that, though, but the fruits, the seeds, the nuts that are produced by those pollination services contribute most of our daily intake of essential vitamins and minerals, as well as the protein and carbohydrates that we get from the crops. So from a health perspective, animal pollination of these crops is absolutely vital. If we focus down to the UK for a moment, the diversity of pollinators within the UK is surprisingly high. They're both phylogenetically and ecologically diverse. And of course, the group that get the most media coverage and the most general interest from the general public are the bees, of course, which is why we have a World Bee Day. We don't have a World Hoverfly Day, unfortunately, though I can see there'll be a lot of support for it. The official DEFRA estimate of pollinator diversity is 1,500 species, and where that figure comes from, nobody really knows. It was written on the back of an envelope one day by some civil servant at DEFRA. But one of the things that we do know is that it is certainly too low. So DOPI, which is the database of pollinator interactions, which is held at the University of Sussex, currently includes almost 2,000 species of pollinators in Britain. And Stephen Falk, whose work some of you will be familiar with, is one of the most experienced field entomologists in the country. He estimates that there's around 6,000 insects that regularly or occasionally visit flowers and may act as pollinators. And if to put that in context, UK has around 24,000 insect species, so potentially a quarter of our insect species are pollinators. And that breaks down into about 270 species of bees, around 60 species of butterflies, perhaps 900 species of larger moths that regularly visit flowers. Similar diversity of hoverflies to the bees, around 270 species of those as well, plus hundreds of species of other flies, beetles, wasps, and so forth. So a really surprising diversity, certainly far more than just bees, and certainly much more than just honeybees. But there are concerns about the diversity and abundance of pollinators in the UK and globally. We're fortunate in the UK that we've got some of the best longer-term data sets on insect diversity and abundance anywhere in the world, and they show a very mixed picture of pollinator diversity and abundance change over time. Wild bees, actually, the latest indicator from JNCC, top right there, which is from 2023, shows that actually wild bees are doing better than most people imagine. There has been an uptick in the last decade or so in their abundance and distribution, which may be due to conservation efforts, though it's a complex picture. In contrast, bottom left there, hoverflies are doing really, really badly. They've really declined, particularly since the late 90s, early 2000s. It's a mixed picture for UK butterflies, which is tracked by butterfly conservation. Habitat specialists are doing particularly badly. Habitat generalists not so badly. But it's Britain's larger moths, also tracked by butterfly conservation, that are one of the real concerns. There's been a 33% decline in moth abundance since the late 60s, and some moth species are around 90% have seen a 90% drop in their populations. Now, the solution to these problems for many of our pollinators is often thought to be, let's plant more flowers, or let's put up some bee hotels, like this enormous bee hotel that I photographed in Paris recently. Things like bee hotels are important, and I wouldn't dissuade anybody from putting one in their garden or on their balcony. Similarly, planting for pollinators is important too, and I wouldn't dissuade anybody from doing that. But it's only part of the story. It's actually a relatively minor part of the story. I think hopefully by the end of this talk, and by the end of Sarah's talk, you'll agree that soils are incredibly important for maintaining pollinator populations. So one of the first bits of evidence to show you to support that statement is based on some work by James Kane and John Neff, who published this paper back in 2011. And in the appendices there, they have some data on the numbers and proportions of what they term mining bees in the different families of bees. This is global data. And by mining bees, we mean solitary bees, mainly solitary bees. Some of them have a primitively used social system, but they're mainly solitary bees. In other words, they don't have workers, they don't have queens, but which create their nests in soil. And I won't go through all of the data within this graph. Suffice to say, the Andrenidae, which is one of the largest bee families, which includes species of Andrena, including this one that I photographed this year in my own garden, 100% of them, or more than 2,500 species, are ground nesters. And if we look at that sort of globally, it's about 64% of all bee species, more than 9,500 bee species, which are mining bees. In the UK, that accounts for about 120 species that rely directly on the soil out of this 245 species of solitary bees. This is data that I took from Steve Falk's Field Guide to Bees of Great Britain and Ireland. And one of the interesting things, and one of the things that we're exploring within the ProPol Soil project, which Sarah will talk about, is the way in which different species of bees show preferences for particular types of soil, particular levels of shade, particular topographies, vertical soil slopes versus shallower soil slopes, and so on. But just to focus on that graph for a moment, if you look on the right-hand side, I've put the 10 or 12 bee species that will use bee hotels. So it's a relatively small proportion of the total number of British bees. Most of them are soil nesters. This figure's from this review by Cecile Antoine and Jessica Forrest from 2021, just really to illustrate how variable ground nesting bee architecture can be. It can be very simple, just a simple excavated channel down into the soil, or it can be much more complex, with mounds of soil that have been excavated, presented around the entrance to the channel, or very, very complex three-dimensional structure within there. So there's a significant excavation of soil within there, and I know that's something that Siul is going to talk about. But as well as that excavation of soil and that turnover of material, there's also an input of organic material as the female bees bring mainly pollen down into the nests, sometimes mixed with nectar. As you'll see in a moment, nests can be very, very deep. They can be constructed up to around 5 meters in depth, and those nest aggregations can be extremely extensive. So again, this is data from the Kane and Neff review, where I've looked at the for individual species the maximum depth that's been recorded for those nests. And as you can see, again, it's broken down into family. Most families and most species within those families have relatively shallow nests, often less than 25 centimeters, and typically within the range 25 to 100 centimeters. But just occasionally you get very, very deep nests described as well, certainly over 5 meters in depth. And recently, you may have seen some media reports about this study by Stephen Hogan and colleagues, which recorded what they think is one of the largest aggregations of a mining bee species ever recorded, a species of Andrena from North America. And this is from a cemetery in New York State, with an estimated population size of between 3.1 and 8 million bees emerging in 2023. So a massive aggregation that has been there for a number of years, and will probably persist long into the future. And I like this figure from the paper on the left-hand side, because it shows us not only their sampling regime and the extent of where they think the nests were, but one of the ways in which they sampled those bees, which is using these emergence traps, which again is something that we're using in ProPol soil. You can trap not only ground nesting bees, but basically any insect that has part of its life cycle on or in the soil. Because it's not just bees that are soil dependent, other pollinators are too. And in fact, if we look across Europe as a whole, clearly bees are very, very important. But actually, most of the diversity of pollinators within Europe, Britain included, are flies. And flies show a significant soil dependency as well. So this is data extracted from Martin Spate's book, his species accounts of European Syrphidae, European hoverflies. And it's a summary from 997 species accounts. And For the 167 species where there was explicit information about the pupae of those flies and where the pupae were located prior to development as an adult fly, almost a quarter of them, 24%, were directly soil related, so that those insects are pupating within the soil. But to me, one of the headline figures from this is over on the right-hand side there. Out of those 997 species accounts, for 830, we had no explicit information on pupal development. Now, that doesn't mean it doesn't exist. It may be there in the literature somewhere, but it certainly wasn't accessible enough to be able to be included within this otherwise monumental report. So that, to me, suggests that there are huge gaps in our knowledge amongst these lesser known pollinators, and that is one of the things that Propol Soil is going to be focusing on. When it comes to butterflies, in Britain, fewer of those are soil dependent at the pupal stage, but certainly some of them are, particularly some of the blue butterflies, Lycaenidae, are associated with ants' nests. So there's a sort of indirect association with soils there. So depending upon how you classify them, there could be as many as 19% out of these 59 species which are soil dependent to some degree. Focus on the moths. Moth data is less easy to parse, but if we look at this pretty important family, the hawk moths, the Sphingidae, which doesn't have a high diversity in Britain, but globally has a very, very high diversity. 11 out of 12 species pupate within the soil, things like these privet hawk moths here, Sphinx ligustri. Almost all of the hawk moths are soil dependent to some extent. So in my 2021 book, Pollinators and Pollination, Nature and Society, one of the things I talked about at some length was that pollinators need more than just flowers. So much of the action that's happening around our efforts to conserve pollinators seems to be focused on planting flowers, maintaining flowers, no-mow-may, for example, agri-environmental flower strips. But pollinators need far more than that. And I developed this idea of the requirements of pollinators triangle. And within that, those requirements, there are three vertices to that. The first of those is flowers. Some pollinators require particular flowers, they're specialists. Others require a diversity of different flower types. They need abundant flowers, and they need flowers that are flowering at the right time, or have a continuation of flowering throughout the season. But the two other vertices, which are often neglected, are reproduction. So, for example, it could be host plants for caterpillars of butterflies and moths, or it could be specific nesting sites for bees or particular microhabitats. And then the third part of the triangle is what I term supplements. These are additional food requirements for those pollinators. So many social wasps, for example, are carnivorous in their adult stage, even if they're visiting flowers to pick up nectar to refuel. There may be particular nesting requirements, nesting materials that are required, or they may need hibernation sites. And we can, just to finish, we can place the importance of soils in all three parts of this triangle. I mean, clearly diverse soils support plant diversity. Some plants are restricted to calcareous soils, so those require acid soils. Some need soils with lots of organic material. Others survive much better in more skeletal soils. Soils clearly support host plant diversity in the same way, but as we've seen, they also provide nesting sites, and many of these ground nesting bees have specific requirements for those nests. And then finally, on the supplement side there, soils provide hibernation sites. So most of our British bumblebee species, for example, hibernate as fertilized queens, as mated queens in loose soil, for example, at the base of hedgerows. But as we've also seen, things like hoverflies and some moths and butterflies also have pupation requirements within soils. So hopefully that's given you an overview, a flavor of the relationship between pollinators and soils, and the complex dependency that pollinators have on soil diversity, soil health. And so now I'm going to pass over to Sara, who's going to tell you a little bit about the ProPol Soil Project. Over to you, Sara.
Thank you very much, Jeff, also for really nicely introducing the topic. And I will just continue. And maybe what is important is that I will pretty much talk about a project. Great. Okay. So yeah, welcome. I think we heard a lot about pollinators at the moment, and this project is something, or yeah, it just started not too long ago, a couple of months. And I think it would never have started if not for the EU coming up and some experts coming up with the idea that we need to understand and manage the soil-dependent pollinators better. And Jeff just pointed out that there's so little knowledge, which we have at this stage, that we really need to do something. But this project also showed that there is like very excited experts who are working on specific topics like soil in the case of soil scientists, and pollinators in the case of pollinator scientists. And we sort of work on these worlds like in our own little bubble. And what this project does, and this is something I'm really appreciating, it brings together two different kinds of worlds. Because I think what is known is, and that's pretty familiar to most of you, soils are, and I just cited here, a scarce and threatened resource all over Europe and beyond, with estimated 60% of soils in the EU being unhealthy due to unsustainable management practices. So this is known. At the same time, we also know that there are soil-dependent insects, and they are just soils under threat because of non-adapted soil management practices. We also know, and I think Jeff pointed it out quite nicely, that pollination is a key ecosystem service, which is highly important for many of the crops that we have and thus for food production. So here are 3 facts that pretty much everyone may know about, but they haven't really been brought together in the past. And I think this is the great thing about the core, which then led to the project ProPol Soil, because it links these hitherto largely disconnected soil and pollinator world. And I just asked one of the LLMs to provide this little icon, because honestly, there is no existing icon that brings together the soil pollinator worlds. I'm not sure if it's the best one that it came up with, but I think it at least shows that this needs to be connected. So yeah, I think you already now know that there are pollinators which depend or even spend a significant part of their life underfoot or in the soil. And bees are just one of them. We saw the hawk moth, some other butterflies. This also applies to hoverflies. Then even things which we don't even think of when we think about pollinators, like thrips, they also spend a large part of their life in the soil, and hardly anyone knows what's actually going on there. And then I think that little guy here is probably also familiar to a lot of people. It's a rose beetle, and they also are actually quite important in the soil. So there's a lot of animals that are dependent on the soil. And this graph just summarizes some of the effects that we are looking at. And again, like the soil world and the pollinator world has looked at some of these factors very independently, but not really connected them. And I just give a few examples. So in the soil, we have a lot of chemicals, some are brought in by human activities. So you have land use, which is bringing in fertilizer, increasing the nitrogen content or the phosphorus content. You also have runoff from industry, also increasing the nitrogen content. And all of this is, of course, affecting the soil itself, but also the biological communities that are living in the soil, such as microbial communities, but also earthworms, nematodes, you name it, like a lot of organisms that are in the soil, including soil nesting pollinators. And we basically don't know how these factors interact, and also how anything else that we do to the soil, so depicted here as, for example, the fact that we are driving on soil with heavy machines, which is compacting the soil, and that may make soil very non-habitable for some of the pollinators that depend on soil as much as for other organisms. So yeah, there's a lot of aspects of the soil world, like the soil biophysics, contamination, also with things like microplastics, pesticides, other toxins that are in the soil that then affect the soil biodiversity, including the pollinators. But as I said, we don't know what's actually going on here. And some of the questions that we are asking are, for example, which role do soils play in pollinator health and which soil parameters matter? So is it pH that matters? Is it the humidity level? Is it how do toxins affect soils, pollinators nesting in soil? So all of these questions are basically unanswered at this stage. And also, how does soil management affect the soil-dependent pollinators? Does it put them at risk? Does it maybe even improve soil at some point? So we really don't know all of this. Or where soil-dependent pollinator populations thrive. So Jeff pointed out this one population. So apparently there, the soil seems to be really great. But we don't know why and what the parameters are. So this is something that we also would like to know, because a healthy soil for pollinator may not necessarily be a healthy soil for other aspects of soil biodiversity, or how we consider it, like as a healthy soil for crop production, for example. And finally, what can we do to improve the habitats that support soil pollinators? And it's exactly those questions that brought us together. So basically experts from the soil world and the pollinator world to start the project Proper Soil. And don't ask me about the name. It was basically the first name that came to our mind. And then we came up with a lot of much cooler names and had a survey within the consortium. And for some reason, we still selected Proper Soil, which is the reason why we have this sort of really hard to pronounce name for the project. But by now it's set in stone, so we use it, and I think we're also quite happy with it. So just a few figures. The project started last year in Freising with our kickoff meeting. So we came together and discussed, like, how are we going to answer all of these questions that I showed you. We have overall 23 partners. You see the countries that are involved in the project here. So it's actually a really nice part of Europe, not all the countries, but I would say a large part of the countries, including some non-EU countries, which is also nice. So we, for example, have Serbia on board, which is really exciting. We also have Switzerland on board. And we have someone from Canada, an expert on soil nesting bumblebees. So it's a really nice group of experts that came together and to work on these questions. And we also have case study areas where we really want to look into management effects on soil nesting pollinators. What's the scope? So we have some aims. We want to gather and share first comprehensive data, like answer these questions on the hidden links between soil and pollinators. We also want to build up long-term hubs that bring farmers, researchers, policymakers, communities together. So I would ask all of you, if you have ideas you would like to support the project, please do, because we really want to reach out and go beyond the scientists. We want to strengthen collaboration also between the EU projects on the soil mission, for example, on soil projects, and the pollinators on the other hand. So really in between the projects. We want to raise awareness, which is hopefully what we're doing at this stage or at the moment, and also include citizens in the citizen science project, and improve how science and policy interact to create a smarter, more coordinated environmental strategies. And just some outcomes over the time, what we want to produce, what should also be useful for stakeholders, policymakers, is like a summary of all the knowledge that is there, which has partly been already put together. Jeff showed some examples, but we are also digging deeper into anything that we may find. So we may have a nice literature review that we can share at the end. We also want to identify these key soil parameters, so which ones are interesting, which ones are important, so that we really know what soil-dependent pollinators need. And we want to use novel monitoring approaches because we really don't have good monitoring approaches to assess the diversity and also the well-being of pollinators in soil, using, for example, new AI techniques or eDNA, which is done in several experiments. And one of the major outcomes is EU maps, which basically show where we have good conditions for pollinators and where we don't have such good conditions for pollinators. And finally, we want to provide information on remediation and mitigation practices. So basically a best practice guide that tells stakeholders what they could do in order to make sure that they manage soil in a way that also supports pollinators. So that's it from my side. I think I could hopefully get you excited about the project, about this link between the different worlds, which I think is very much needed. And if you want to support us, I can only point out this citizen science project that we have. It's an iNaturalist project, where if you know of pollinators, particularly bees that nest somewhere in the ground, somewhere close to where you live, you can just sign up, take photos, provide us with the data, because we really need a lot of data from a lot of people to better understand what's going on here. So yeah, that's the big group of people involved, and I think we are even growing because we are just recruiting more PhD students and postdoctoral researchers. So it's a big group which has a big task, and we are very much looking forward to working on this project and to hopefully provide much more insight on the exciting world of soil nesting pollinators. Thank you very much for your attention, and I just leave this for a second and then hand over to the last speaker.
Hello, can you guys see presentation? Yeah, hope so. So yeah, my name is Siul Ruiz. I'm gonna shift gears a little bit and focus a little bit more on the soil and the health of the soil and the structure of the soil, obviously from a biophysical perspective. And my background is on soil bioactivation, particularly with worms and plant roots, but extending this to ants. And here we're going to also talk about mining bees as well. So this elusive word, soil bioactivation, it's actually the biological process of structure formation by these different biological agents. And that is the movement and displacement of soil particles by flora and fauna. And it's also, if you look at the inverse image, it's the pore structure that results from this organization. But here I'm only talking about mineral movement. We also have the mixing and redistribution of soil organic constituents and injection of soil organic constituents in the form of carbon. And all this plays a role for a healthy soil. So this serves several ecosystem services. For example, flood mitigation, so providing drainage pathways. It extends oxygenated depths of soil, so you can have enhancement of biodiversity for these oxygenated depths. Stimulation of microbial activity, so for example, rhizosphere soils are very different than, let's say, barren soils. And the structure itself provides paths of least resistance for subsequent biological activity. So for example, root growth or other faunal movement below ground. So the actors that we're talking about here are often deemed ecosystem engineers. So some of the more famous ones that you might have heard of, the humble earthworm, but we also have plant roots that modify their environments as well. We have larger mammals that operate in a scale quite different to that, which I've been talking about thus far. We've got minor bees, we've got termites, we've got ants, there are reptiles, there are arachnids. So it's a large list of actors. And these actors have strategies that are adapted to various constraints. So you've got biophysical constraints. So whether you have a soft cell membrane versus a more rigid cell wall, you have an exoskeleton versus a hydroskeleton, which may determine the mode of operation that you're behaving under. But we also have climatic constraints, so wet regions versus dry regions, cold regions versus warm regions, and other environmental constraints such as soil texture, soil moisture, or the hydromechanical characteristics of that soil. And we also have, for example, chemical characteristics such as soil pH. So here, what I'm going to be discussing in particular are two operators, or two categories of operators: penetrators, which mechanically generate tunnels by penetration and cavity expansion. So earthworms and plant roots are the examples that I'm going to be using here. And then I'm going to talk about excavators, those that use their limbs or mandibles to carry or shovel out soil masses, such as ants or bees. And the reason I make these distinctions and strategies is to try to paint this beautiful picture of different regions and how some of these strategies have been adapted to certain conditions in these regions. And then at the very end, I'm going to throw a little bit of a wrench into this whole painting by going into a little bit more depth about the burrowing bees and discuss the nuance and certain constraints that fall categorically between the conventional excavator and the conventional penetrator. Now, obviously, this is not exhaustive, but it's just to, as we said before, give a flavor of the importance of this discussion. So when I talk about penetrators, here I'm looking at plant roots and burrowing earthworms. So plant roots operate by generating cells at the root tip. That cellular division and sloughing near the tip creates lubrication at the forefront. Some of those cells get reoriented through an enzymatic cutting, and they then end up behind the root tip and extend axially just behind, causing this penetration. Now, earthworms, on the other hand, they're constructed, well, to say constructed is a little bit harsh, but they have circumferential and longitudinal muscles, which they can synchronize in contraction and flexion in order to produce a peristaltic motion. That peristalsis enables them to anchor locally and extend tip-wise, creating a penetration cavity expansion motion. So obviously, physiologically, there are huge differences between these two agents. For example, I'm going to describe this as actuation speed, which is actually just extension speed by the plant root. They're on the order of 0.1 to 0.2 microns per second, whereas earthworms operate 3 orders of magnitude faster than that. But like I said earlier, the stiff cell wall enables plant roots to exert pressures on the order of a megapascal, which is an order of magnitude higher than that of earthworms, which lies somewhere between 100 and 230 kilopascals. So physiologically, they're both very different, but they both perform a very similar mechanical behavior, which is penetrating through the soil. So we can describe that physically using the process of cavity expansion. So if we treat the soil as the unsaturated soil as an elastoplastic body, we know the material properties that constrain this activity. So soil shear strength, which is effectively a yield stress, and shear modulus rigidity, which is a sort of elasticity. Those are both moisture and texture dependent. We have a system of equations, basically our equilibrium equation, which is effectively Newton's third law, a response to that, which is the deformation and the deformation gradients, and constitutive laws here. So you'll see we've got a plastic flow law, and when we're in the elastic regime, we've got the elastic response. Using some simplified assumptions, we can come to an analytic solution that says this is the amount of pressure required to create a biopore, and that is a local inelastic term, which is the failure around the penetration regime, and then a remote elastic term, which bounds the problem. Okay, so I have this analytic solution. We know soil mechanical properties are dependent on moisture and texture. We can then actually look, me and a colleague actually, we looked at soil maps and looked at wetter regions versus drier regions. We looked at coarse versus fine textured soils, and we generated a map. And what this map is saying is, based on the earthworm's maximum pressure, we can say where we likely see earthworm activity and where we probably shouldn't be seeing earthworm activity. When we compare this with occurrence data, we have a 90% agreement with our maps and the occurrence data. We have some false negatives. Those are usually in areas that don't have high enough resolution, so near riverbanks where you actually can see the river, but obviously we're not able to predict the moisture at those resolutions. But importantly here, we can also show temporal windows. So using a monitoring site near Southampton or in the New Forest, we actually were able to look at the weather, make predictions about the amount of pressure required to punch a hole in the soil. And we can see the earthworm abundance actually drops when the pressure gets too high, when it goes above that hydroskeletal pressure limit. So what I want to say here is there's no sophisticated statistics here. There's no machine learning. It's just understanding the physics behind this process. Now, the take-home message for this is worms are more active under wetter conditions and finer textured soils. That is in a slight contrast to the ant story or the excavators in the broad brush strokes when we talk about excavators. So here I'm going to be talking about ant tunnels and bee nests. They create these similar kind of turrets, and depending on the complexity, they have these nests with compartments and structures. But the process is entirely different. So now if we look at the ants, ants actually operate based on granular displacement of large, almost discrete soil particles, either aggregates or grains. And the constraints here are the mandible size, or what they can actually carry, the aggregate size, so the dimension of the aggregate, and the soil particle size. And obviously the weight is also a limiting feature here. But the particle size informs what kind of forces are at play. So whether it's largely capillary forces, or if we have van der Waals forces that actually keep these particles or these aggregates together. And to skip all the details here, what we find is that the granular displacement is actually facilitated by coarse textured soils because they're large discrete units readily available for being carried, and drier conditions because aggregates, even in finer textured soils, stay bound under drier conditions and they're lighter in weight. There's less water in them. Those are exactly the conditions that would impede earthworm activity. And what we see here is taking a richness map. So red, the color map is a little bit skewed here. Red is bad for worms up here, good for ants down here. You can see that there's ant activity where you won't find earthworm activity. Likewise, you'll find earthworm activity where you see reduced ant activity. So the point is that these strategies fill certain niches that the other cannot. Obviously, there's overlap as well. But we can start to paint this picture and this tapestry of soil bioactivation. And now comes the wrench. We add the bees into this topic. So where do the bees come in? So the minor bees, they are actually, like as stated before, often solitary bees, but they generate this soil structure. They are excavators by nature, but their process is a little bit more nuanced. See, the minor bees will actually fly away from their nest upwards of 5 meters to take in water into their gullet. They'll use that water, they'll spill it onto their burrow surface at the forefront to weaken and soften the soil. So they actually need the soil to be soft in order to excavate the soil. And then they'll dislodge masses of soil. They'll roll it up into a sort of a ball or aggregate, and then they'll actually remove it. And that's how they actually operate. And then it's rinse and repeat from here. They'll actually go through that cycle over and over, which obviously this must cost energy. There must be certain limitations to it, which we don't entirely know. But the construction does create a sort of nest, which may have this sort of interweaving network of tunnels, which again facilitate movement of water, oxygenated depths. But they also have these compartments that I actually use a soil plug to block off. So those compartments actually, that's where they store their nectar and pollen, and they actually have their eggs and their youth in there. So when it rains down too hard, even if the rest of the nest becomes infiltrated with water, these soil plugs should hopefully preserve these areas so that they don't get too damaged. So that's the below ground story. Obviously, pollinators have activities above ground as well. And this is something that we're just now starting on. So above ground, there seems to be an interaction between bees and plant roots. So some of the working hypotheses, we just won some funding to actually start monitoring this activity from an acoustics and vibrations perspective. And one of the working hypotheses here that we want to actually test is that the bees actually generate either acoustic or vibrational waves that trigger the plants. And though we don't know the physiology behind this exactly, that plant response is to produce nectar to actually attract the bees so that they actually come forward, take the nectar, and aid with the pollination. Now, there are other questions as well. Can the plants actually produce sounds on their own? Are they actually detectable sounds? Do they act to attract the bees? So all of these questions we don't have answers for as of right now, but they are within the remit that we're going to be studying in terms of vibrations and acoustics experiments, theoretical monitoring or theoretical modeling, and biological inferences as well. So this work, this grant that we're just now starting, is to gain a more holistic view of the pollinator plant interactions, the phytoacoustics generated by the plants, and the below ground activity. So the ecoacoustics associated with soil bioactivation and those interactions. So just to kind of finalize here, I know I've spoken quite fast recently. Understanding these processes helps us to predict specific agents for specific regions. Understanding the fundamentals enables us to identify bioactivation activity windows, which will help us to inform when that structure is going to be formed. And importantly, it'll help us to make predictions about, in case climates are supposed to change in the future, anything like that, how these pictures might be changing and which agents might be taking over or handing off the baton to another. Now, obviously, there are plenty of nuances and unknowns. For example, when we look at the minor bees, we don't entirely understand all of the physical limitations, the energetic requirements, the mechanical requirements, and everything in the middle. So these nuances have to be better understood in order to paint this more holistic picture and in order to understand what the interactions and the feedbacks actually are. And then if we can understand this, we'll better understand how we can actually generate soil structure and recover that soil structure after, for example, the case of soil compaction, or even worse, something such as wildfire recovery, because you're not going to necessarily see ants readily invading a wildfire damaged site. Okay, so as I stated before, these are just the first steps in painting this tapestry, and I hope I've given you a really rough flavor of the importance of soil bioactivation in the context of soil health and degradation recovery. And these are just quite some of the acknowledgements I wanted to make, including Paul Hallett, who's here. The project is actually run by Mikhail. I'm a co-I on this acoustics project with Mikhail Kokovsky. And yeah, this ends my talk. Thank you very much for having me here, and thank you for your time.
Great, thank you very much to Siul and also to Jeff and Sara. I know Jeff has to head off right at 1 o'clock. We're going to keep the question and answers open beyond 1 o'clock, but I appreciate a lot of people attending. We'll have to leave. Just a quick question to Jeff to kick things off. In your talk, you mentioned there were 9,500 species of minor bees, which absolutely blew my mind. If you look at across these species, do they all have the same burrowing mechanisms in soil, or are they quite different?
That's a really good question. The short answer is I don't know, but I imagine that they are rather different because there's a big diversity of different soil types that they require. I know Siul will know more about this than me, but I can imagine that burrowing into very sandy soil requires a different mechanism compared to burrowing into a clay soil or an organic soil. But also the structure of the nest, the architecture of the nests differ enormously. and the positioning of the nests, whether they're vertical or whether they're on shallow slopes or steep slopes. I can imagine all of that would require different approaches to constructing those nests. But yeah, there's a huge amount we don't know about that, as Sara was saying.
All right, and I've got various questions that people have submitted onto the Q&A box. Please keep submitting those as well. I'm going to pass another one to you, Jeff, just because I know you're going off early, but I'll get Sara and Siul to also talk on this. So what effects do you think tillage is going to have on ground nested pollinators, particularly given we've had a big shift towards either minimum tillage, where just the top few centimeters get disturbed, or even to no tillage, where we don't disturb the soil at all? So let you answer that first, Jeff.
Yeah, again, complex responses depending upon the groups concerned. I mean, for those bees which are shallow nesting, for example, tilling could be enormously damaging. But then you've got to think about the timing of when that tillage happens. Is it happening before or after nest construction? The interaction of tillage with or non-tillage with the use of pesticides and herbicides as well. I know that in many non-tillage systems, they're encouraged to use herbicides to destroy the vegetation that's the crop that's there, and then plant into there. So that may have a greater effect on the pollinators and on the insects than the tillage itself. Yeah, no, I'll pass on to the others, see what they have to say about
Sara,
that.
do you have anything you'd like to add?
I think there's not much I can add to what Jeff already said, but I think maybe Siul has another idea from this biophysics perspective.
Yep. So on biophysics, go on, Siul.
I'm not entirely sure. I know there was recently an article that came out looking at the impacts. I think it was an opinion article looking at the impacts of soil compaction caused by tillage machinery. And I think it's still just an ongoing study, given the current, given the current understanding of the biophysical interactions. It would seem that they would be subject to some constraints. They're not quite. It's unclear how strong they necessarily are and how resilient they would be to compaction. And again, those kind of shallower nests are probably going to be a little bit more susceptible than deeper ones. But yeah, I think it still remains a little bit, from a mechanistic perspective, an unknown.
Okay, and next question, any of you can answer this one, but it's from Konstantinos, and it asks that considering that most of the literature on nesting depths of ground nesting comes from the United States, how different do you expect them to be in colder climates like the UK? Who would like to answer that?
I've got my microphone on, so
Okay, go ahead.
So that was specifically about depth, yeah?
It's about depth, but I think you can answer it more broadly if you'd like to.
Yeah, I mean, it is going to vary geographically, definitely. I mean, bee biogeography is complex, so you have families which are only found in some parts of the world, not other parts of the world. As we've mentioned, the type of excavation differs, so I can imagine that there'll be huge differences there. Whenever we do broad biogeographic assessments of pollinators or plant pollinator interactions or pollination webs and so on, there are always massive geographic gaps in our knowledge, particularly in parts of Africa, particularly in parts of Asia as well, and often in areas where there's very, very high diversity of bees and other pollinators. So yeah, I'm sure there are enormous biases, if you like, within that data.
Okay. Anyone else like to comment on depths? What might happen if it's colder? There's probably going to definitely be Siul?
Maybe
Well, Sarah.
Oh, sorry. No, it's as I said, it's probably a lot of educated guessing here, because exactly for the reason that we pointed out, there's so little known. But I think this is where I really appreciate it to listen also to the talk of Siul, that there are general probably biophysical properties, and even though that's from, I mean, it's still a temperate region, so there might be things that we can learn from it, and we'll definitely use it. But then again, like, we do have this very like things that work at a very micro scale, because also besides the soil properties, you also have a different different interactions of whatever climate you have, which is of course driven by various factors. So I think this is what makes it so hard. So one of the tasks that we have is producing these maps. And just for Europe alone, without having super detailed information, we will probably never produce like maps that go to a very low level of resolution, because exactly for this reason. So we sort of may have a broad idea, but we can also use the data from other parts of the world. But if they are 100% correct is really hard to say. I think it just gives us sort of like an educated guess of how the situation may be at a specific spot. This is a bit how I see it. So it's useful, but it will not provide, we'll never be able to provide the detail that would be nice to have.
Yeah, just to follow up on what Sarah said, with regards to the speculation, I mean, I would imagine there's going to be difficulty, reduced, what is it? You know, when it's under colder conditions, you're going to have a reduction in activity, generally speaking, for biological activity. Potentially deeper burrows might help with handling or dampening some of the temperature changes, but yeah, I guess you're going to have reduction in activity, generally speaking.
Great, thank you. Jeff, I think you need to get to another meeting, and maybe some of the attendees do as well. If Siul and Sara are okay to stay on, I'll ask a few more of the questions. Yep. So thank you very much.
Yeah.
Talk.
Thanks, Paul. Thanks to all the attendees and my fellow presenters. It was very, very interesting. Bye-bye.
Bye, Jeff. Excuse me from the meeting.
Oh, well, yeah, send your apologies. Yeah.
All right, I'm going to go down to a technical question from Mutsar Ali, and he sounds like someone who both of you should probably get in contact with, because he uses plaster of Paris to excavate complete bee nests from underground. But if he does this, he finds that the soil breaks and it's very difficult to do. So he's also tried metal containers and other things to bring out the soil structure. I want to hear the answer to this as well, because I think I'm going to have similar challenges. So for both of you, can you mention your expert opinion regarding how you can successfully observe bee nests underground? And I'm going to pass to Siul first on that one.
I'm not sure. I would say if you manage to excavate some subvolume, it's probably worth. If it's thin enough, you might want to try neutron radiography, because then you might get some information about where moisture might be distributed, where that might lie, and that might even infer information regarding organic constituents distributed within that soil mass. We've done, also Paul, done work with X-ray computed tomography. So this might enable some structure, some detail regarding the structure formation. But again, that's going to be a snapshot, so it's not going to be super dynamic, but that might be the cleanest way to get some structure information with minimal disruption, but it's still going to be pretty disruptive.
Sara, anything to add on that?
I think I can just add that this is a really difficult task, and I've never done it myself. I never even tried, but just trying to summarize the things that I learned from experts who did it or who tried it, and who do experiments with bees. I think it's super difficult to really excavate nests. I think what they sometimes do is that they try to dig buckets into at spots where we know that there's nest aggregations. And I think some of the stories is that, of course, they're always nesting outside of the bucket. But sometimes they manage to get them in the buckets and then bring them to the lab. But I think it's a very, frustrating, time-consuming exercise. And part of what we want to do is probably also improve the possibilities here. But at the moment, I don't have a good recipe. If you find one, I think we'd be more than happy to learn.
Okay, and I'm going to go to the last question for the webinar. It's from Annette Raffin, and it is quite a good one. It's, do you think that ground nesting insects might avoid areas with earthworms on the basis they might damage the nests? So I'm going to pass that to Sara first.
Can you repeat it, Paul? I didn't fully understand it.
Basically, do bees avoid areas where there's earthworms because the earthworm bioturbation would end up damaging the bee nest?
That is a great question. The answer is I really don't know. It's unfortunate Jeff isn't here because he did more of the literature research. He might have we might have known, but I don't. I wouldn't want to make any predictions here.
I reckon no one's ever studied that, but it would be interesting to know what earthworm populations are next to pollinators. Siul, any?
I obviously don't know the answer to that, but what I can say that's interesting is there was a study some time ago by Ivan Kapowitz, which was looking at almost like the psychology of earthworms as they were coming to a new burrow. So if they see, or an old burrow, if they see a burrow from another species, they're a little bit more likely to go into it. But they were quite avoidant when they come to an earthworm burrow that wasn't the same species. And it wasn't clear exactly why that was, but they were significantly more avoidant when they were coming to a burrow of an earthworm of another species. I don't know if that actually translates to when they come to a bee nest. I have no idea, but this is just something odd about behavior of earthworm activity. So
excellent. I'm just going to mention one of the questions that was in here because Jeff answered it by text. And it was on VOCs and the fact that we know that above ground flowers attract pollinators with VOCs. I think you're looking at this in your new project, Siul, but is anyone looking at soil VOCs and how they may attract pollinators that anyone knows of?
No, I don't think so. I think Jeff mentioned exactly the experts or the colleagues who are working on this, but I don't think that anyone has looked at the interaction of soil volatiles and plant volatiles.
Is that something you'll look at in your acoustics project, Siul?
It wasn't immediately what we were going to look at, but there is actually some part of our group that look at rhizosphere soils. So it could be on the table actually to investigate whether or not these interactions have a feed forward or feedback to the pollinators. So this is potentially in scope.
Okay, excellent. All right, well, thank you very much to all the presenters. This is the end of the webinar. I'm sorry we've gone over time slightly. So thank you very much, Siul. Thank you, Sara. And we've already thanked Jeff as well. So on behalf of the British Society of Soil Science, I want to express our thanks to all of you for attending today as well. And you'll find a quick feedback survey when you leave the webinar, and hopefully you'll have the time to complete that. The recording of the webinar will also be available after the event on our website and also on our YouTube channel. And the next webinar is scheduled for the 4th of June, and further details will be on the British Society of Soil Science website and via follow-up email as well. Hopefully we'll see you at future events, and in the meantime, thank you again and goodbye.
Thanks, Paul, for organizing.
Thank you.